Evening viewer, welcome to our Glory Globe TV. It's breaking news. Yes, a research team at the Riken Center for Biosystems Dynamics Research in Japan has succeeded in establishing a model of the blood-brain barrier using modularized tissue derived from human cells. The tissue in a cube is a small cubic structure that could provide a boost in the drug discovery field and be used as an alternative to animal models in preclinical studies. The study is published in Communication Biology. The blood-brain barrier is a strict gatekeeper around the brain that prevents foreign substances in blood from entering the brain. Although protective, the barrier poses challenges when treatments need to affect the brain in order to work. When developing drugs for treating brain diseases, it is thus crucial not only to test the drug's effectiveness inside the brain but also to confirm that the drug can pass through the barrier. Developing new drugs is time consuming and costly at the early preclinical stages. Conventional methods rely on animal testings. However, differences between animals and humans sometimes make it difficult to predict a drug's effectiveness in humans as well as any harmful side effects. Along with increases in number of regulations governing animal experiments, these problems have encouraged researchers to develop research methods that do not rely on animal testings, such as using organoids, structures that mimic human organs, as well as organs on a chip, with the ultimate aim to create artificial human organs for external use. With this in mind, the research team led by Masaya Hagiwara at Riken BDR has developed a new model of the blood-brain barrier using the cube-type system that they recently established for modularizing different human tissues. Reconstructing multiple tissues simultaneously and analyzing their interactions is extremely challenging because drugs have to traverse different types of tissues before reaching the target area, but also crucially important. In the case of the blood-brain barrier, drugs must pass through vascular endothelial cells, astrocytes and pericytes before they can enter the brain. To construct the blood-brain barrier model, the researcher created 5 mm cube frames, filled them with hydrogel embedded with astrocytes and pericytes derived from the human brain, and seeded vascular endothelial cells differentiated from human-induced pluripotent stem, that is called iPS cells, onto the surface to form cell sheets. They then tested the device according to Hagiwara. We are happy to find that is accurately mimicked the real blood-brain barrier in terms of structure and function, with astrocytes and pericytes extending three-dimensionally beneath the sheet of endothelial cells, and there, like the real barrier, it only allowed limited substances to pass through. Behind the report, Glory Globe TV, Delhi, Bharat.